Today we'll be starting off with chloroplast. Can you tell me what are chloroplasts? What is their function? What are the structure of the organelle? What do you think?
Um, Chloroplasts contain chlorophyll and they're the site of photosynthesis, and chlorophyll is a green pigment in chloroplast.
Exactly. They're double membrane structure, only present in plants. And we have seen its electronic structure in the very starting figures. We talked about the structure. What was it like? We have these thylakoid membrane sacs. This one thylakoid membrane sac is known as what? Grana. It is known as grana. And a lot of these thylakoid membrane sacs are known as grana as well. So these thylakoid membrane sacs inside a double membraned organelle is the site where the chlorophyll is actually present. Chloroplasts tend to have an elongated shape and a diameter of about 3 to 10 micrometer. And comparing 1 micrometer diameter for mitochondria, like mitochondria, they are surrounded by two membranes which form the chloroplast envelope. The main function of the chloroplast is to carry out photosynthesis. And during the first stage of photosynthesis, which is the light dependent stage, light is absorbed. Now, when you'll come in A2, you'll study at chapter number 13, which is photosynthesis. In A2, we are going to talk about that there's a light dependent stage and then there's a light independent stage, which does not need light. Now, the light dependent stage obviously takes place in the thylakoid membrane sacs where the chlorophyll is already present, where it's absorbed by the photosynthetic pigments, particularly the chlorophyll, and the pigments are found on the membranes of the chloroplast. The membrane system consists of the fluid-filled sacs called thylakoids, which spread out like sheets in 3 dimensions. In places, the thylakoids form flat disc-like structures that stack up like piles of coin. Now, yeah, it stacks up what like how? A pile of coins. These are how these thylakoid membranes are actually present in a chloroplasts. And these thylakoid membrane sacs contain the chlorophyll which is needed for photosynthesis. Got it?
Yeah.
So what is thylakoid? A flattened membrane-bound fluid-filled sac, which is the site of the light-dependent reactions of photosynthesis in a chloroplast. The second stage of photosynthesis is the light-independent stage, which does not use light. We use the energy. Now, how do we get energy if in this stage we are not using light? The detail will be in your chapter number 13. But you should know that energy comes from the reducing powers when we convert carbon dioxide into sugars, and there's a whole Calvin cycle that you'll study in A2. There's a whole Calvin cycle which is going to tell you how this light independent stage takes place. Plus, we also have a lot of energy that is stored as starch grains. And we have lipid droplets as well, which appear in the electron microscope. These lipid droplets also provide us with energy. Can you tell me if plants store their glucose in the form of starch? How do animals store their glucose?
Uh,
In the form of glycogen. Okay,
oh, yeah.
good. Like mitochondria, chloroplasts have their own protein synthesizing machinery and they have 70s ribosomes. So how do we know what was the endosymbiont theory that we studied yesterday?
Uh, that mitochondria and chloroplast evolved from prokaryotes
And what was the proof for that? That's what I'm asking. Yeah.
because they have 70 as ribosomes.
And prokaryotes also have 70s ribosomes till date, all bacteria. So this means that chloroplast and mitochondria evolved from a bacteria. Got it? Prokaryotes.
Yeah.
They also have a circular DNA because they all have the same machinery which is needed to make proteins. And in your chapter number 6, you'll study where and how is the DNA, ribosomes, RER, all these machinery, rRNA, mRNA, how are they all used to make a single protein? In electron micrographs, the ribosomes can just be seen as small black dots in the stroma. As with mitochondria, it has been shown that chloroplast originated as endosymbiotic bacteria and in this case, photosynthetic blue-green bacteria. The endosymbiont theory is discussed in more detail in the earlier section on mitochondria as well. Next, we have cell walls. Can you tell me what are cell walls and where are they present And what is their function?
Cell walls are like they're made out of cellulose, and they are like they make the plant more rigid, and they give the plant structure,
Hmm,
and so that it doesn't like unlike animal cells, if it has too much water, it'll burst, but plant cells won't burst because it is cell wall.
good. The first walls formed by plant cells are known as primary walls and they are relatively rigid. The primary wall consists of parallel fibers of the polysaccharide cellulose running through a matrix of other polysaccharides such as pectins and hemicelluloses. Cellulose fibers are inelastic and they have a very high tensile strength, meaning that they are difficult to break by pulling on each end. This makes it difficult to stretch the wall. For example, when water enters the cell by osmosis, the structure of cellulose is described in chapter 2. In most cells, extra layers of cellulose are added to the first layer of the primary wall, forming a secondary wall. And in a given layer, the cellulose fibers are parallel, but the fibers of different layers run in different directions, forming a cross-ply structure. Now you can see over here how are they, and in biological molecules, our next chapter number 2, we'll study the structure of cellulose in detail. That how are they all arranged in a crisscross manner. Now this crisscross manner is all actually going to make it very, very strong. Got it? Which means they
Yeah.
would be difficult to stretch, difficult to break, difficult to change shape as well, and having a high tensile strength. So here you can see the two chloroplast and all of that. Some cell walls become even stronger and more rigid by the addition of lignin. What is lignin?
It's present in like xylem vessels, and
Yes,
like it's it's like it makes it more uh like
strong.
rigid so that it
Yeah,
won't like
exactly. You studied this in
collapse.
IGCSE that xylem have lignified walls. The same is the case with cellulose as well. Xylem vessel elements and scrillin chyma, which we'll study in your chapter number 7, they add to the compressional strength to the tensile strength, and it is what gives wood its strength, and it's needed for transport in shrubs and trees as well. Now, what are the functions of cell wall? Number one, mechanical strength and support for individual cells and the plant as a whole. Lignification is one of the means of support, and turgid tissues are another means of support that is dependent on strong cell walls. Cell walls prevent cells from bursting by osmosis if cells are surrounded by a solution with a higher water potential. Moreover, different orientation of the layers of cellulose fibers help determine the shapes of the cell as they grow. So, keeping them rigid, keeping them in shape. Then the system of interconnected cell wall. Now, you should know that these cells are like connected like this, very close to each other. Now, there is a thing known as plasmodesmata that we'll study in chapter number 6 and 7. These cell walls are also very helpful in transporting. There's a thing known as apoplast. Apoplast is a pathway that is taken from cells. It's a pathway that the water takes from cells, and how does it travel from one cell to the other with the help of those roots or those channels that are present in the cell walls as well. Living connections through neighboring cell walls, for example, the plasmodesmata, these pores. This is what plasmodesmata, yahan pe, which is something holding the two cell walls together. They help form another transport pathway through the plant known as the symplast. The cell walls of the root endodermis are impregnated with severin, a waterproof substance that forms a barrier to the movement of water, thus helping in the control of water and mineral ion uptake by the plant. Got it?
Yeah.
So what are the functions different? Can you summarize all these 6 functions?
It prevents the cell from bursting if the cell has a lot of water. It provides strength and rigidity for the cell, and it allows the cell to connect with other cells to the plasmodesmata, and um yeah,
Good.
and it's waterproof also.
Good job. Vacuoles. What are vacuoles?
It's like a sac inside the cell that's filled with water and ions.
Exactly, it's a sac that filled with water, ions, food, storages, enzymes, hormones, pigments, colors, substances. It's like a storage place. ठीक है? As we have seen, and can you tell me the difference in vacuoles inside an animal cell and in a plant cell?
Animal cell vacuoles are smaller and there's multiple vacuoles. The plant cells just have one big like vacuole.
Yeah. As we have seen, animal cell vacuoles are relatively small and include phagocytotic vacuoles, food vacuoles, and autophagic vacuoles. Unlike animal cells, plant cells typically have a large central vacuole, and some examples of the functions of the large central vacuoles of plants are listed below. So, for example, number one is support. How do you think support is provided by a vacuole in a plant cell?
It like expands and then it makes the plant cell more turgid.
How does it expand?
Like it absorbs water.
Good job. Exactly. That's what I wanted to hear. So it's written, the exact same thing is written. What do we call that? What there's a term for the expansion.
Not sure.
Turgid. The solution in the vacuole is relatively concentrated, which means concentrated means what? A high water potential or low water potential?
Low water potential.
Exactly. So when vacuole has a low water potential, water is going to enter and it's going to build up the pressure inside a vacuole. A fully inflated cell is described as turgid. We call this stage turgidity. Turgid tissues help to support the stems of plants that lack wood, and wilting demonstrates the importance of this as well. Wilting is like when leaves fall off. Okay, why is that? Because their vacuoles are not functioning. Then lysosomal activity. Plant vacuoles also contain hydrolases. Hydrolases are the enzymes that are present in lysosomes as well. Then secondary metabolites. A wide range of chemicals are also there inside the vacuole. So vacuole is a storage organ as well. It has those hydrolases as well. It has all those chemicals which are not that essential, but they are important for growth and development, and they contribute to survival in various ways. These are often stored in vacuoles, and examples of their functions are, for example, anthocyanins, which are the pigments responsible for most of the red, purple, pink, and blue colors of the flowers and fruits. They also attract pollinators and seed dispersers. Got it?
Yeah.
Certain alkaloids and tannins deter herbivores from eating the plant. Latex is also present in vacuoles. Now, what does latex mean? For example, if you've seen, there's a white fluid that comes out of the leaf when you tear it apart. That milky fluid accumulates in these vacuoles as well. And this fluid is also there where, so the insects don't eat up the leaves as well. And that fluid is also used to create many drugs as well, such as opium and heroin. Moreover, food reserves. Where does the food come from, which the vacuole is storing?
Um, like the glucose, like the starch.
Where did the glucose come from?
Photosynthesis.
Exactly. Photosynthesis takes place, glucose is formed, glucose is converted to starch, starch is stored in vacuoles. Got it? Then colors as well. Color of the petals, color of the beetroot, color of vegetables, waste products. What do we mean by waste products? For example, any idea?
Like oxygen and
Good,
photosynthesis.
exactly. Excess oxygen produced as well. And growth in size. It also helps in growth because if you look at a diagram of a plant cell, the 90% of the area is taken by whom? Vacuole. If the vacuole is going to grow, then obviously the whole cell is also going to grow as well. And that is all due to the osmotic uptake of water into the vacuole, which is responsible for most of the increase in volume of the plants. All right. What is this? There are therefore shuffle the cards, start and okay, let's do this activity. Okay, how do we start? Okay, we have to match, I guess. Okay, you're going to tell me the correct answer and I will match. Why is it start? Why is start written over here? And यहाँ लिखा हुआ है. Let's start with chloroplast. Can you tell me its function? The options are
Uh,
given over here. Tell me.
this one.
Exactly. ठीक है. Nucleus.
Uh, make ribosome.
No. Nucleus. What is nucleus?
It controls cell activity, but that's
Yeah.
not the.
Chromosomes are found in nucleus. This is a
Oh,
nucleus. Chromosomes, you forgot? Chromosomes. Chromosomes contain what? DNA, remember? Second
yeah.
organ. Ribosomes,
Uh, found on the rough endoplasmic
cell wall.
contains cellulose.
You know what? They're actually written all above. ये सारे ऐसे ही आंसर लिखे हैं. All the answers are like this. Nucleolus makes up the ribosomes. Mitochondria, ATP synthesis. Golgi apparatus makes lysosome. Cilium has this arrangement of microtubules, and lysosomes contain digestive enzymes. It was actually a game that we're supposed to cut out, and then we're supposed to play that game as well. Okay.
Oh, okay.
Yeah, well, I guess it wasn't a joining game, but still we had to practice. Let me show you one thing, then we'll start with bacteria. Can you tell me one general thing? What is the difference between bacteria and viruses? One main major difference.
Here are cells, but viruses are not considered cells. So
Mhm. Viruses are not considered as cells.
yeah,
Good. And what is the reason for that? Why do we say that viruses are non-living things?
I think it's because it like needs something else to like live. Like
Exactly.
it's like
The host's
it need-
body. Correct.
yeah,
Okay. It only can live inside, reproduce, respire, carry out nutrition, all activities, move, breathe. Every activity, every characteristic is carried out once it is inside a host's body. So now we'll start off with bacteria. Is everything clear since the start, unit number 1.1 to 1.6? All clear?
yeah,
Are you revising as well daily?
yeah.
Okay, now we'll start off with bacteria. You will recall that there are two fundamental types of cells, prokaryotes and eukaryotes. Give me examples of eukaryotes first.
Plants and animals.
Plants, animals, yeah, exactly. Prokaryotes, bacteria. The plants and animal cells you have studied so far are the eukaryotic cells. Bacteria are prokaryotes and their cells are much simpler than those of eukaryotes. Prokaryotic cells are generally about 1,000 times smaller in volume and lack a nucleus that is surrounded by a double membrane. The very first difference between a prokaryote and a eukaryote is that eukaryote have a DNA which is enclosed inside a proper nucleus, but prokaryotes have a free DNA. It's literally free. It's not even enclosed inside anything. Okay? It's not even enclosed inside a membrane. Nucleus is a proper double membrane structure in eukaryote. This is eukaryotes and prokaryotes. It's a free lying DNA. Prokaryotes are thought to be have been the first living organisms on Earth. The earliest known fossil prokaryotes are about 3.5 billion years old, and most biologists believe that eukaryotes evolved from prokaryotes about 2 billion years old. There are two groups of prokaryotes that you have to keep in mind, bacteria and archaea. Now, what are the different structures that are always present in all bacteria? A cell wall, and the difference in the cell wall of a plant cell and a bacteria is that cell wall of a plant is made up of cellulose. I'm going to write down some materials. These are very important for MCQs. In plants, the cell wall is made up of cellulose. In bacteria, the cell wall is made up of peptidoglycan. And can you tell me about fungi's cell wall?
Chitin.
Exactly. How do you remember that?
We did this in IGCSE, I think.
Good. These are the three very important MCQs for your yes. Okay. So cell walls would always be present in all bacteria. Cell surface membrane would always be present, cytoplasm, and a circular DNA would always be present. Now, all bacteria have these four structures. Now you can see the DNA is lying free in the cytoplasm. It's not even enclosed inside a nucleus. It doesn't have a membrane or it doesn't have a nucleus or anything. Sometimes some bacteria, for example, let's take an example of Vibrio cholera bacteria, which causes cholera. Now, Vibrio cholera bacteria or some bacteria may also contain a flagellum for locomotion. What is locomotion?
What?
Locomotion.
Uh, like
Movement.
moving?
Moving, exactly, simple moving. Some bacteria also have a capsule or a slime layer, which is an additional protection. Then some bacteria also have these infolding cell membranes, and some bacteria also have a plasmid. Plasmid is also a small circle of DNA where many important characteristics of the bacteria may be present. Then we have pili, which is involved in sexual reproduction and binary fission. Binary fission is to be studied in A2 level. Binary fission can that these pili play a very important role because this is how bacteria reproduce and create more in numbers. Is the structure clear?
Yeah.
So we have seen that the left side of the diagram shows the structures that are always present in bacteria, and the right side of the diagram shows the structures which are sometimes found in bacteria. Bacteria are a group of single-celled prokaryote microorganisms, and they have a number of characteristics such as the ability to form spores, which distinguish them from the other group of prokaryotes known as archaea. Now they have a cell wall which is made up of peptidoglycan, and the cell wall protects the bacterium, and it is also essential for the survival. It prevents the cell from swelling up and bursting if the water enters the cell by osmosis. And peptidoglycan is basically a polysaccharide which is combined with amino acids, and it is known as murin. Murin की cell wall बनी होती है. Murin is made up of these peptidoglycan chains. It makes the bacterial cell wall more turgid. Then the second structure was the cell surface membrane. Like all cells, bacterial cells are surrounded by a cell surface membrane. It is a partially permeable membrane. And what does a partially permeable membrane mean?
It doesn't let all particles in, like it controls the movement of particles.
Exactly. Cytoplasm. The cytoplasm does not contain any double membrane bound organelles such as mitochondria. But if we talk about a cytoplasm of eukaryotes, that cytoplasm would have many double membrane organelles such as nucleus, mitochondria, chloroplast, which are not present in prokaryotes. Then circular DNA is a very important structure. The DNA molecule in bacteria is circular and it is found in the region called a nucleide, which also contains proteins and small amounts of RNA. It is not surrounded by a double membrane, and unlike the nucleus of eukaryotes, there may be more than one copy of the DNA molecule in a given cell. Ribosomes, on the other hand, what is the function of ribosomes?
Protein synthesis.
Yes, good job. Protein synthesis. And how are the ribosomes in eukaryotes and prokaryotes different?
Eukaryotes is ATS and prokaryotes is CAIE.
Good. Flagellum. What is the function of flagellum?
Allow movement.
Allow movement, exactly. They are able to swim because they have one or more flagella as well. Bacteria flagella have a much simpler structure than a eukaryotic flagella. The bacteria flagellum is a hollow cylinder made up of identical protein molecules. We studied yesterday, na flagellum किसका बना होता है? Microtubular structure ही है basically. It is a rigid structure, so it does not bend, unlike the flagella in eukaryotes. It is a wave shaped and works by rotating at its base like a propeller to push the bacterium through its liquid environment. As a result, the bacterium moves forward with a corkscrew-shaped motion. Then the different infoldings inside the cell surface membrane, they are a site for biochemical reaction. You don't need to study the detail, but that why are these cell surface membrane inner folded? You just have to remember that they are very important for some biochemical reactions such as aerobic respiration and all of that, because they also contain photosynthetic pigments which allow photosynthesis to take place as well. Then nitrogen fixation you'll study in A2 is a very important bacteria as well, which eukaryotes do not contain. What is the function of this capsule? Any idea? Capsule is there for extra protection. Some bacteria are surrounded by an extra layer outside the cell wall, and this may take the form of a capsule or a slime layer. A capsule is a definite structure made mostly of polysaccharides, and a slime layer is more diffuse and it is easily washed off. Both help to protect the bacterium from drying out and may have other protective functions as well. For example, a capsule helps protect some bacteria from antibiotics, and some capsules prevent white blood cells known as phagocytes from engulfing the disease-causing bacteria. Is it clear?
Yeah.
Plasmid, very important. This will also be studied in detail in A2. That this is a small circle of DNA. Now you are saying, why is it not part of this circular DNA? Why is it a separate one? This is a separate one because it contains some specific characteristics which are very unique to each of the different bacteria. Plus, it has many functions as well. The genes the circular DNA contain is a general one, but the genes that this plasmid actually contain have various useful functions. Commonly, plasmid contain the genes that give resistance to particular antibiotics such as penicillin, and they can copy themselves during binary fission as well and can spread rapidly from one bacteria to another. Whereas the circular DNA has all the basic characteristics that are present in normally all of the bacteria. But the plasmid is a special DNA, you can say, which has the various essential and important functions. Last structure, which was the pili. Pili are the fine protein rods and they vary in length and stiffness. 1 to several hundred may be present on the outside of the cell, and they are used for attachment and interactions with other cells or surfaces. They allow the transfer of genes, including plasmids, from one bacterium to another during conjugation. Got it?
Yeah.
Are the 10 structures of bacteria clear? You're going to write them all today in one liner. You have to explain me all in the class and you have to write them down as well. Let me show you a differentiation between eukaryotes and prokaryotic cells. Okay, where is it? Yeah.
And we're back in third quarter planning. Adams is quarterbacking this project, and all living things are made up of
cells. There are many different types of cells. For millions of years, prokaryotes were the only.
So what are the two types of prokaryotes that they have named?
Bacteria and protozoa.
No. Protozoa is a separate group. Prokaryotes contain bacteria and archaea.
Oh.
Yeah, listen carefully. This is very important.
For millions of years, prokaryotes were the only form of life on this planet. Eukaryotes came later as a result of the process of evolution. Another difference between eukaryotic and prokaryotic cells is their size. Eukaryotic cells are generally larger than prokaryotic cells. Eukaryotes are mostly, although not entirely, multicellular organisms, whereas prokaryotes are always single-celled, or unicellular organisms. Examples of unicellular eukaryotes include amoebas, paramecium, and yeast. The structure of the DNA in eukaryotic cells is different from that in prokaryotic cells. In the nucleus of eukaryotic cells, DNA forms tightly bound and organized chromosomes. Prokaryotic cells contain just a single loop of stable chromosomal DNA stored in the nucleoid. The nucleoid is not a structure, but the area where the DNA is found. Both types contain ribosomes, but in eukaryotic cells they are bigger and more complex and bound by a membrane. Most eukaryotes reproduce sexually. The offspring have genetic material that is a combination of the parent's genome. Prokaryotes, however, reproduce asexually. Their offspring are clones of the parent cell, which come about through binary fission. Finally, prokaryotic cells have a larger surface area to volume ratio than eukaryotic cells, which results in a higher metabolic rate, and therefore increased growth rate and shorter generation time. While eukaryotic and prokaryotic cells are quite different in their structure and processes, they do share similarities. Ribosomes are one feature they have in common, but both also have a cell membrane composed of phospholipids and proteins. The membrane provides a barrier between the external and internal environments of the cell and selectively allows certain materials to pass through. Both types of cell have DNA as the basis for their genes, although the structure is different. The genetic material regulates cell function and contains the coded information that is passed on to offspring. Both also contain cytoplasm, but in eukaryotic cells, it is defined as everything within the cell outside of the nucleus. In prokaryotic cells, the cytoplasm refers to everything contained inside the cell membrane. The gel.
So what are some of the differences that you can see in both of the structures, and can you highlight some of those and tell me?
So eukaryotes have nucleus and membrane-bound organelles, but prokaryotes don't. And then prokaryotes have wait, hold on.
One difference you have already told that about the DNA, okay, which is enclosed inside a eukaryote, but it's a free DNA in prokaryote. Any difference in the cell wall?
Like bacteria cell walls made out of peptidobacter, and then plant cell walls made out of cellulose.
Good. What about the flagellum and pili?
So prokaryotes have flagellum, but eukaryotes don't.
Good. Good. Okay.
Like cytosol is a major part of the cytoplasm in.
Okay. So let's go through comparing both the prokaryotes and the eukaryotic cells. Prokaryotes are thought to have evolved about 3.5 billion years ago, and eukaryotes are thought to have evolved about 1.5 billion years ago. The typical diameter of prokaryotes is 1 to 5 micrometer, and cells are up to 40 micrometer diameter and up to 1,000 times the volume of prokaryotic cell. If we talk about the DNA, what was the difference in DNA of prokaryotes and eukaryotes?
Prokaryotes is like RNA and eukaryotes is DNA.
No. Prokaryotes DNA is circular and free. Eukaryotes DNA is inside a nucleus. Got it?
Oh, okay.
Yeah. Next is 70S ribosomes in prokaryotes, but 80S ribosomes in eukaryotes. Then in prokaryotes, very few types of cell organelles are present, and they don't have any membrane bound. For example, in eukaryotes, we have many different variety. Some of them are single membrane, such as lysosome, Golgi apparatus, vacuoles, and endoplasmic reticulum. Some of them have two membranes as well, nucleus, mitochondria, chloroplast, and some of them have no membrane as well, ribosome, centriole, microtubules. So we have a whole variety in eukaryotes, whereas there's no variety in prokaryotes. And what's the difference of cell wall?
Bacteria has peptidobacter, but eukaryotes, plants have cellulose, and fungi have chitin.
Good. Then flagella are simple and they lack microtubules. They project outside the cell surface membrane in prokaryotes, but over here they are only arranged in microtubules inside the cell. Cell division occurs by binary fission when we talk about prokaryotes, and cell division takes place by mitosis or meiosis when we talk about eukaryotes. Some prokaryotes carry out nitrogen fixation. None of the eukaryotes carry out nitrogen fixation. So this is a very, very important table which has 9 differences highlighted between prokaryotes and eukaryotes. Is it clear?
Yeah.
Good.